Welcome class. So today we'll be looking at computational biochemistry. In this lecture series we'll look at how to predict the protein function, how we can determine the function of a protein, the domains, the different domains of the protein, the transmembrane region. For example, those membranes that span across membranes. So how do we determine which part of the amino acid, which part of the protein segment that make up that are embedded in the membrane? And we're also looking at the secondary structure of protein using Ramachandran plot. So in the secondary structure, we look at the alpha helical structure of the beta plated sheet of protein and how we can determine if the protein exists in the alpha helical state or in the beta plated state. So this is actually a protein. So we'll look at the function. There are softwares which you can use to determine the function of the protein. So we have the ProtParam. The ProtParam is actually a web-based software that we can actually use to determine the physical-chemical properties of the protein. So in this case, okay, let's see if we can navigate the internet. I just clicked on the link. So in this case, we have this is a tool where we can paste the FASTA sequence of the protein. The FASTA sequence is just the alphabetical, each amino acid have a different alphabet. So we look at how we can get the FASTA sequence. Usually we get the FASTA sequence from the Ncbi. So we have the Ncbi, the National Center for Biotechnology. So this is Ncbi. Okay, this is Ncbi. But I've actually downloaded a FASTA sequence which I'm going to use in this case. So the FASTA sequence I have here, which I actually saved on my notepad, I have different proteins here. I have the hemoglobin, the beta unit of hemoglobin. So I'm going to copy it. I'm going to copy the FASTA sequence here. Like I told you, each alphabet denotes a particular amino acid. So I'll copy this, then I'll go back to my ProtParam. Let me say, sorry, I'll go back to my ProtParam. So you can see very well. This is my ProtParam. Sorry, I think I'm using the web base. So I've pasted the sequence here, compute parameters. So this will give me the user provided sequence. So this is amino acid 1 to 10, 11 to 20, 21 to 30 to get to this 140, this 141, 42, 43, 44, 45, 46. So this is total number of 146 amino acids. So here we have the documentation, which you have the number of amino acids and this particular protein, which is the hemoglobin. So we have 146. The molecular weight, this is molecular weight, which is 15,867.22 molecular weight. So we have the theoretical isoelectric point, which we have the 6.8. Okay, this is the amino acid composition of hemoglobin. We have the alanine 10.3%. We have the arginine, the asparagine, the aspartate, the cysteine, glutamine, glutamate, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, thyroxine, valine, and phenylalanine. So now if you look at this, okay, these are the different composition of the amino acid, and this is the percentage composition of the amino acids. Now here we can also predict if the protein contains more of essential amino acids. For example, they might actually give you a protein source for people that have protein energy and nutrition. You can actually, if this is the protein of choice for the nutritionist, you will now look at if this protein contains essential amino acids. Look at the percentage amino acids, so we can determine if it's a first class protein or a second class protein. Depends on if the protein contains more of essential or non-essential amino acids. So here, but this is not the focal point. The focal point of this lecture is to look at the amino acid composition, the physical chemical properties of the protein. Now you can see we have total number of negatively charged residues, which is the negative charged amino acid, which is aspartate and glutamate. So we have 15. Yeah, we have total number of positive charged amino acids, which is arginine and lysine. We have total number of positive charged 14. So in this case, what I have, this is the atomic composition, total number of carbon, hydrogen, nitrogen, oxygen, sulfur. So this sulfur here, we have 3 here, shows that our protein contains some sulfur containing amino acids. Example of such amino acid is the cysteine and the methionine. So if you look at the composition again, let's see if we can see cysteine. Okay, yeah, we have cysteine, 2 cysteine. Do we have any others? Okay, only 2 cysteine. So do we have methionine? Okay, this is methionine 1. So 1 plus 2 giving us what? 3. So that makes the total number of sulfur in this protein to be 3. This is the molecular formula, total number of atoms, extinction coefficient. The extinction coefficient tells us the absorption properties in water for this one, while this one tells us the absorption properties, when you look at, assuming all pairs of cysteine residues form cysteine. What is cysteine? 2 cysteine residues forming the disulfide bridge. That is what we call the cysteine. So the molar extinction coefficient, assuming all pairs of cysteine residues form cysteine. Here, in case of assuming all cysteine residues are reduced, they have SH. In this case, they have SH in this case, not just like cysteine to cysteine. So if it is reduced, it means that cysteine is standing alone, but if it is oxidized, that means it is forming a kind of cysteine-like structure. So here we have the estimated half-life. Okay, we have the instability index. This actually tells us how stable our protein is in the test tube. So you can see this classifies the protein as stable. So we have classified this as being a stable protein. So we also have the aliphatic index, which tells us the aliphatic nature of the protein. So we can also use the okay, let me just, please do not forget to subscribe, share and comment for more lectures on bioinformatics, protein structures and termination. Thank you.